Hi, everyone. Thank you for joining this session. I'm Andres Diaz-Pinto. I'm part of the TME team at NVIDIA. Today, I'm very happy to share with you briefly, show you the latest work that I've been involved, which is on surgical robotics and digital twin technology. Just to give a bit of a background where this project is located. So we are currently working on systems that help surgeons during the operations. They are also called robotic surgical assistants, or RSAs. Definitely, I see a lot of potential. We see a lot of potential on this assistant changing the way we surgeons basically do their work. Based on the literature we read and the work that we, the test that we did, we found that the surgeries using assistants make the surgeries more precise and definitely help to reduce the fatigue that the surgeons experience during long surgeons. So essentially, our goal is to improve surgeons' dexterity using AI and simulation in Omniverse. And to achieve this, we use the behavior cloning technology, which is an approach to train policies or to train models based on expert demonstrations. So these are the steps that we follow. So essentially, the first step is to create photorealistic content, photorealistic anatomical models based on synthetic CT volumes or real CT volumes. And this is where Michael said that we use Macy, and I'll briefly describe how we use Macy in the next slide. But yeah, so this is the first step, to create the content and in OpenUSD format. So we use the NVIDIA Omniverse to the simulation. Once we create the photorealistic content, we compile a teleoperation, or we gather a teleoperation data set for 5 different tasks, which again, I'll describe later as well. And finally, we train a couple of state-of-the-art models on these demonstrations. Specifically, we use the action chunking transformers, or ACT, and diffusion policies on the demonstrations that I'm going to show later. So let's discuss a bit of how we create the photorealistic content, or based on the CTs. So here is the diagram, or basically bench of steps showing the 3D workflow that I follow to create the photorealistic content. As our first step is basically create synthetic CT scans using Macy. And then once we synthesize the CT volumes, we segment the organs, which is the second step, right? Just to highlight here, so this workflow uses 2 inputs, actually, or can use 2 inputs. So one is the synthetic CT volumes, or real CT volumes, in case we want to plan a surgery and visualize that in Omniverse, right? So the inputs are actually, or can be both a Macy, a synthetic CT volume, or a real CT volume. For the real CT volume, we can use Vista 3D to segment the different organs, 100+ regions, or also 3D sec, right? Okay, so once we segment the organs, we obtain the masks, which is the second step, right here. You can see the organ segmentation here. You just create a GIF showing how the segmentation looks like. But then we did a couple of steps before we move to the next step, which is we add semantics to the labels. And the idea of doing that is that because we have 100+ regions, we didn't need that amount of say detail. By that I mean, for instance, the vertebrae, which is around 24 or 25, depending on the person. We merge all of the vertebrae and create one region which is called spine. And the same we did for the ribs. So we didn't need to have independent ribs. So we just merge all the ribs and have a single region. The same for heart and the same for the lungs. Again, this step is to simplify manipulation and the analysis of the anatomical structures. The third step is converting the masks, the organ mask, into OBJ files. And this is basically because it's the standard 3D file format for visualization or for any software that uses surface meshes. The next step is to clean the meshes, and for this we use ZBrush. And the whole idea of this is to remove any imperfections or artifacts in the meshes, and in general to create better UV maps, which is the input to the next step to basically to create photorealistic materials, right? So once we clean the meshes, we move to the next step to create the textures, and for this we use Adobe Substance Painter. So the input of that was the UV maps. And once we had the UV maps and the textures, we assembled everything in OpenUSD file, which is the file format that we use for simulation in Omniverse. So let's now talk a bit more about the experiment setup that we use for all the simulations and teleoperations. So we use Orbit Surgical, which is the framework that was designed for surgical robotics, and is based in Isaac Lab. And the great thing about Orbit Surgical is that it simulates the Da Vinci Research Kit, or the DVRK. It models joint articulation, low-level controllers, and it allows us to replicate the physical behaviors of the DVRK. Also, one of the great things about Orbit Surgical or Isaac Lab is that it also provides high fidelity rendering, so we can tap from the best rendering algorithms in Omniverse, which is crucial to develop vision-based algorithms, right? For the visual data, we use the camera sensors to capture 512 by 512 pixel RGBD images, which means color and depth information, right? And we also use a wrist-mounted camera to mimic the surgeon's view when doing the surgery. Okay, so this is the experiment setup. So let's now discuss a bit of how we collect the teleoperation data set. So for each of the tasks that I'm going to mention in the next slide, we collected 50 expert demonstrations through human teleoperation, which is in total like around 7 hours of demonstrations. And we recorded this demonstration using VR controllers, which provide us 6 degrees of freedom, and it basically mimics the controls of the DVRK gripper in the 3D space. And I want to highlight the importance of using the VR technology here, which help us to capture all the nuance actions of the expert surgeons. These are the tasks that we mostly focus on. So the first one are 2 simple tasks, which is tissue retraction and needle lift. And for tissue retraction, we basically make the robot to use so basically the gripper to grasp and lift the tissue. And success here is when we achieve the robot to correctly grasp the tissue and at a specified location, right, and lift it. That's basically the goal of this. And for the needle lift it's very, very simple. The name says it. So it's straightforward, but it's critical, right? So it basically makes the gripper to go to a position where the needle is and lift it, right, to a specified height. So that's the second simple task. The other, a bit more complex, are the needle handover, switcher path, and the block transfer. The needle handover is so we have 2 grippers, right? So it's a 2-arm operation. The right or the left basically grasp the needle and move it to at a handover point. So then the other gripper just takes over the needle and move it away to the position where the gripper took it. In the suture path task, basically the robot grasps the needle and thread it to the center hole of the suture path. And here, success is basically when the needle passes completely through the opposite side, right? And I'm going to show you videos of how the models perform on all these tasks. For block transfer, it's just moving a surgical training block from one place to the other, right? So that's the goal of the block transfer tasks. So briefly, let me discuss a bit of the models or the architecture that we use. So the first one, or one of the architecture that we use, is the action chunking with transformers, or ACT. And basically, this architecture integrates a conditional variational autoencoder with a multi-head attention transformer. And this, based on past observations, the ACT predicts sequences of actions which are called or named action chunks. And the idea is that these multi-step actions makes the ACT effective for tasks that require coordinated movements, right? So it's one of the state-of-the-art architecture used for behavior cloning, right? And I basically put the reference here for you to read more about the ACT architecture. The other one was the diffusion policies, which we use to train the models based on the demonstration that we created. And here the key difference between ACT and diffusion and ACT is that the diffusion, instead of predicting a single action or a sequences of actions directly, these policies start with a noisy sequence and progressively refine it, right? So that's the idea. It's like sculpting, right? So you start with a rough block of actions and then iteratively refine it until you have a smooth and coherent sequence. And again, I'm not providing much details of this, but I put the reference so you can go and check more details of these architectures. So now let's have a look at the success rates that we achieve across all different setups and tasks. So you can see here tissue retraction, needle lift, needle handover, suture path, and block transfer. And you can see the results that we got, the success rate for the different setups for single camera, multi-camera, using point clouds for ACT and for DP3. So we generally observe that DP3 outperforms ACT. So you can see success rate for most of the tasks are usually mostly higher than ACT. So we also saw that for simple tasks like tissue retraction and needle lift, the success rate was quite high. For tissue retraction, which is a simple task, was almost 100% successful. But then for the more complex ones, you see that it's a bit more difficult to have the model or the policy to do all the tasks 100% accurate. But still, it's very good results considering the amount of observations that we use to train those models, which is only 50, right? It is nothing to train a policy actually. We want to just to compare, how the ACT and the DP3 performs on these different tasks. One thing that I want to highlight as well is the generalization, specifically on needle lifting. So we train a policy on one type of needle, the needle 1. So you can see the shape here, and we test the model on different shapes or different needles and see the success rate for the different shapes of the needles, right? We found that using multi-camera on the action champion transformers was the best performing one. So you see the success rate when we use the same needle is 70%. But then when we change the needles, you see the performance drop, but still is quite high compared to the other configurations. And we wanted to make this experiment to see how the model generalizes on different, setups or different, specifically different shape of the needles. Also, just to finalize and just open the door to questions, I just wanted to show you some of the manipulation tasks, some of the examples of the policies trained and how they perform on the different tasks. For instance, for the tissue retraction model, here is the model, the train model lifting the tissue. This is not a teleoperation. This is the actual model doing the task. You can also see the needle lift. And this is the two of the more complex tasks, which is needle handover and the switcher pad. So we see one gripper taking the needle and then moving the needle to a handover position. So the other gripper take it and move it to another position. On the right, you see the switcher pad model passing the needle through the switcher hole. And this is quite impressive. I mean, this is not the teleoperation video. This is a model performing the task, the train model performing the task on 50 demonstrations, right? So just want to highlight that. And the last task is a block transfer model. So we put the block in a random position, and the gripper take the block and move it to a specific location that we want.